In this experiment, we tested the effects of 3 different drugs, adenosine, histamine, ADH, on the properties of the flow of fluid through blood vessels and across the capillary wall. This was done through analyzing the weight change, which represented the exchange of fluid across the capillary wall, and the drip rates of the frog, representing the flow of fluid. Clinically, adenosine is used to treat arrhythmias. Drugs with histamine are involved with inflammation causing allergic reactions. Antidiuretic hormone, also known as vasopressin, can be used to increase blood pressure and to reabsorb sodium. All these can affect the blood vessels of the body, causing vasodilation or vasoconstriction. In terms of methods, we use the same setup as the fluid balance experiment done earlier in the quarter. We produced different solutions containing adenosine, histamine, or ADH through the frog's aorta to study the effects of the different drugs. The flow of fluid through the vessels, represented by a drip rate, is represented by Ohm's law. Ohm's law describes the flow through a tube. In our experiment, the tube was the vessels in our circulatory system. Ohm's law states that the flow of fluid through a tube is equal to the pressure difference along the tube divided by the resistance. The resistance is based off of multiple parameters, which include fluid, viscosity, length, and radius of tube. Of these parameters, only the radius is affected upon drug application, causing a change in resistance and therefore a change in flow.
2,000 years later.
The movement of fluid across the capillary wall between the frog's circulatory system and interstitial tissue was represented by the change in wave. This could be described using Starling's law of ultrafiltration. There are 3 parameters within Starling's equation that affect the direction of fluid movement. These are the forces that drive fluid from the tissue into the capillaries, the forces that push fluid out of the capillaries into the tissue, and the filtration coefficient. There are 2 forces that bring water from the interstitial tissue into the capillaries. The first is the interstitial hydrostatic pressure, or P sub I. This is the pressure inside the tissues that push water into the capillaries. The second force is the capillary oncotic pressure, or pi sub C. This is the pressure generated from the solutes within the capillaries that attract water inward. Of the forces that push water into the interstitial tissue, there are 2 components. The first is the capillary hydrostatic pressure, or P sub C. This is the fluid pressure within the capillaries that is pushing water out. The second force is the oncotic pressure of the interstitial tissue, or pi sub I. This is the pressure that's like an osmotic pressure that attracts water towards it and is generated by the solutes in the interstitial tissue. The last of these 3 parameters is the filtration coefficient, or K. This represents how effective the water can cross the capillary bed and is dependent on the size of the tight junctions. For this experiment, this value was assumed constant.
Before adding adenosine, a controlled drip rate and weight change was taken. The weight change was 25.8 milligrams per minute for the control, and when adenosine was added to the ringers and dextrin perfusate, the rate of weight change increased to 120.4 milligrams per minute. The control drip rate was 10 drips per minute and increased to 12 drips per minute with the addition of adenosine. Adenosine acts on the A2 receptors, which are located on the membrane of the frog's smooth muscle cells surrounding the arterioles. When adenosine binds to the A2 receptor, the G-alpha-S signaling cascade is activated, and the alpha subunit of the associated G protein breaks off from the trimeric protein. This alpha subunit binds a GTP molecule and becomes activated, and this leads to an activation of adenylate cyclase, abbreviated AC. AC is an enzyme that converts ATP into cyclic AMP, or CAMP. Cyclic AMP activates protein kinase A, abbreviated pKa, which then phosphorylates a number of targets. pKa will phosphorylate potassium channels, activating them to pump potassium ions out of the cell. This will repolarize the cell membrane, and this repolarization acts as a negative regulator on the L-type calcium channels, or the LTCCs. The LTCC is a voltage-gated calcium channel on the membrane of the smooth muscle cell, and when inhibited, will prevent calcium influx. pKa will also phosphorylate the myosin light chain kinase, abbreviated MLCK, and the myosin light chain phosphatase, abbreviated MLCP. When phosphorylation occurs, the MLCK becomes inactivated and the MLCP becomes activated. The activated MLCP will remove the phosphate group off of the myosin, while the inactive MLCK prevents further phosphorylation of the myosin. Net effects of adenosine binding the A2 receptors are as follows: increased potassium ions leaving the smooth muscle cells, and less phosphorylation of myosin. Because the phosphorylation of myosin heads is a crucial step in muscle contraction, the lower levels of phosphorylated myosin equate to less smooth muscle contraction. This will result in vasodilation of the arterioles. Vasodilation increases the radius of the tube, and from Ohm's law, this decreases the resistance within that tube, ultimately leading to increased flow through the circulatory system and an increased drip rate. Also, arterial vasodilation means that downstream there is more fluid in the capillary, increasing the capillary hydrostatic pressure. From Starling's equation, more fluid was being pushed out into the interstitial space, increasing the weight of the frog.
Many months later.
Before adding histamine, a controlled drip rate and weight change was taken. The weight change was negative 104.1 milligrams per minute for the control, and when histamine replaced the Ringer's dextran perfusate, the rate of weight change increased to 5.3 milligrams per minute. The control drip rate was 10 drips per minute and increased to 12 drips per minute with the addition of histamine.
Dumb. Girls is dumb.
Sandy! Okay, Sandy, I get it!
What's that? You want more? Okie dokie. More water for the sea critters.
Okay, Sandy, okay! I get it!
No more.
Histamine acts on the H2 receptors, which are also located on the membrane of the frog's smooth muscle cells. Once histamine binds to the H2 receptors, the G-alpha S pathway again becomes active. From here, the same signaling cascade as seen with adenosine occurs, leading to vasodilation of the arterioles and an increase in the drip rate and weight of the frog.
Before adding ADH, a controlled drip rate and weight change was taken. The weight change was 40.8 milligrams per minute for the control, and when ADH was added to the Ringer's and dextran perfusate, the weight change decreased down to negative 197.1 milligrams per minute. The controlled drip rate was 12 drips per minute and decreased down to 10 drips per minute with the addition of ADH.
I don't need it. I don't need it. I definitely don't need it. I don't need it. I don't need it. I don't need it. I don't need it.
I need it!
ADH acts on the V1 receptors, which are found in high density in the vascular smooth muscle membrane. When ADH binds to the V1 receptors, the G-alpha Q signaling cascade is activated. Once again, the alpha subunit breaks off and it binds to GTP, activating it. This complex then leads to phospholipase C activation, or PLC. When PLC is activated, it hydrolyzes and splits PIP2 into two secondary messengers, diacylglycerol, or DAG, and inositol triphosphate, or IP3. DAG is fat soluble, and it stays in the membrane and will later increase protein kinase C activity. PKC phosphorylates and activates the L-type calcium channels, which allow calcium influx from the extracellular fluid into the cytosol. PKC can also act on and turn down the function of potassium channels. This prevents potassium outflux and slows down the repolarization of the smooth muscle cell. IP3 is water soluble, and it enters the cytosol where it will bind to the IP3 receptor on the sarcoplasmic reticulum, a calcium storage protein, and this opens it up and allows calcium to flow into the cytosol. Overall, the net effect of DAG and IP3 is a higher concentration of calcium ions in the cytosol.
A few moments later.
Calcium is a trigger for smooth muscle contraction, So this increased concentration in the cytosol results in more smooth muscle contraction in the arterioles. The mechanism for this contraction is seen through calcium's binding to calmodulin, or CAM. The calcium calmodulin complex activates myosin light chain kinase, or MLCK, which phosphorylates the myosin heads. Once myosin is phosphorylated, its ATPase becomes activated, and the myosin-actin cross bridges and power strokes can now form. This ultimately results in contraction of the smooth muscle. The vasoconstriction decreases the radius of the arterioles, which increases the resistance within the tube and therefore decreases the flow of fluid. This decreased fluid flow is represented by the lower drip rate. Constriction of the arterioles also meant that less fluid was entering downstream at the capillaries. This reduced volume of fluid caused a decrease in the capillary hydrostatic pressure, which resulted in less fluid being pushed into the interstitial tissue. Therefore, the net flow of water movement was from the tissues back into the capillaries. This led to a decrease in the weight of the frog.
So much later that the old narrator got tired of waiting and they had to hire a new one.
In conclusion, the addition of adenosine and histamine led to vasodilation of the frog's arterioles through the G-alpha-S signaling cascade. This resulted in increased drip rate and increased weight of the frog. ADH caused vasoconstriction of the arterioles through the G-alpha-Q signaling cascade. This resulted in a decrease in drip rate and a decreased weight of the frog.